Good evening. I don't know how many of you have ever attempted to hold your breath for a minute or more. Those of you who have, I think you would agree with me that it is extremely unpleasant. And that is because our bodies rely on oxygen to survive. Under optimal conditions, the level of oxygen in the environment equals the demand of oxygen from the cells in our body. Those are shown by those blue arrows. So the oxygen enters our cells and into these little factories called mitochondria that can use this oxygen to generate energy in the form of ATP. And this ATP is what fuels everything that we do, our muscle contractions, our ability to signal to our different cells in our brain. And so it's absolutely essential. And given that oxygen is so important, when there is an imbalance between the amount of oxygen available, shown by fewer oxygen molecules there, but the demand of oxygen remains high, this causes a crisis in the cell, where essentially the cell cannot produce enough ATP to power what we need to do. And oxygen that is missing in the environment might be one cause, but in addition, there are cells and tissues in our body that may simply not get enough oxygen delivery to operate at an optimal level. And so this can lead to a variety of diseases, either causing these diseases or exacerbating these diseases because there simply is not enough energy to function. So despite this, there are many organisms that have found a way to adapt to low oxygen. And I'm showing them here. They're called hypoxia-tolerant organisms. And one of these organisms that my lab studies is called the zebrafish. The zebrafish is remarkable in the sense that it can survive for up to 50 hours in complete absence of oxygen. And so it's a great model for trying to understand how nature can do without this essential element. So while all the organisms that I showed you there are hypoxia tolerant, they do it different ways. But there are also some conserved mechanisms, meaning mechanisms that occur across all of these organisms. And those are the molecules that my lab is particularly interested in identifying because if we also possess them, that means that if we understand how they work, we may be able to modulate them to mitigate hypoxic injury. So to give you an example of such a conserved molecule, I'm showing you here the hypoxia-inducible factor called HIF-1 alpha in blue. It's a molecule that is extremely unstable under normal oxygen conditions. It gets tagged, and that causes it to be degraded in essentially the cell's garbage disposal, which is called the proteasome. However, when oxygen is low, these tags are removed, and the level of HIF-1 alpha can accumulate. And what happens next is that these molecules can enable other molecules that have important adaptive functions to be expressed. For example, vascular growth factors increase, enabling more blood vessels to be formed. More red blood cells are also generated. And there are other molecules that enable the cell to make energy in a mitochondria-independent manner. So essentially, this all enables cells to do the best that they can with very little oxygen. And so this discovery was seminal because it was one of our first strong pieces of understanding of how essentially cells can adapt to a very harsh and changing environment. And it led to the Nobel Prize for these three scientists in physiology and medicine in 2019. And building on that work, there are currently now clinical trials for drugs that either activate or inhibit HIF-1 alpha to serve for treatment for anemia and cancer. So my laboratory is interested in identifying other molecules like HIF-1 alpha. And we are going back to these hypoxia-tolerant organisms because they are able to induce other kinds of responses that are probably more extreme and enable organisms to survive under extremely harsh conditions. These conditions are such that you have very little oxygen, but what these organisms can do is they can reduce the demand for oxygen, shown by that one little blue arrow there. And so what this means is that while the supply is low, the demand has also decreased, and the organism reaches a new state of stability. And it can function by preserving ATP. So in our attempt to discover molecules that might induce this state of arrest, if you will, we performed a screen with the expectation that any molecules that are important for survival will be upregulated under these very harsh conditions. And so we performed a genetic screen and identified a molecule that has a terrible name. I didn't choose it. It's called NMYC downstream regulated gene 1 alpha NDRG1, and it's shown there in red. And this remarkable protein can associate to other proteins, the sodium potassium ATPase, which in subsequent slides I'm going to call NKA. This is an enzyme that uses a lot of energy to carry out its function. And so it's not at all optimal to keep this thing going when oxygen is low. And on the other side, NDRG1 can interact with other proteins or molecules that transport target proteins to various compartments of the cell. So what we have shown is that in low oxygen, the levels of lactate elevate, and NDRG1 can grab this pump and associate with trafficking proteins and deliver this pump again to the cell's garbage disposal. And we believe that this is a method by which cells can conserve energy under extreme situations. So I want to walk you through a few experiments that helped us to demonstrate this model. So the first thing we did is we wanted to determine where this molecule is expressed, NDRG1. And we found that it is in the kidney and the ionocytes. And these cells serve the common purpose of maintaining ion homeostasis. And this is a very energy-demanding process. So from there, we were wondering, what might this molecule be doing? And we decided to go through the database for zebrafish embryos and to find other molecules that might have a same spatial distribution. And we found the sodium potassium ATPase. They have remarkable similar distributions seen here with different labeling methods. And as I told you, this pump is very energy demanding because it has to pump sodium and potassium against their gradient. What we further demonstrated is that under low oxygen, the levels of NDRG1 are high, and NDRG1 binds to lactate, whose levels increase under low oxygen. NDRG1 can then bind to the sodium potassium ATPase pump and lead it to the cell's garbage can. So this is all very well and good, and it seems like there is an interesting correlation, which doesn't mean causation, between the association between these proteins and the degradation of the pump. So we believe that this is a method for the cell to, as I said, preserve energy. And the last experiment that I'm going to show you is one that actually demonstrated this more directly. We were able to get rid of this molecule, NDRG1, and we observed to our satisfaction that these fish looked perfectly normal. They grew to adulthood, they were fertile, but they died at a much higher rate than normal fish when exposed to low oxygen. So we then looked at the levels of sodium-potassium ATPase in normal fish and fish that were lacking this protein, NDRG1. And what we saw is that the levels of NDRG1 go down over time in normal fish, but remain at a very high level in fish that are lacking NDRG1. So this led us, this is quantified here, wild type in the black line and the mutant in red. And so this led us to conclude that NDRG1 might be a molecule that can turn on a low power mode in the cell. And that in this low power mode, organisms and cells are able to endure quite a long period of oxygen deprivation. And so NDRG1 is a molecule that we also have. And we think that the only difference between fish and humans is how this molecule is regulated. So where my lab hopes to go in the future is to find a way to activate NDRG1 independently of low oxygen. Because with the ability to do so, we could enable cells to be protected when oxygen levels drop. So I know I'm a little bit over time, but I'm the last speaker, so I'll say one last thing. And that is that, help me dream here, I think that it would be possible in the future to take an organ that is waiting to be transplanted. That organ sees very little oxygen and sees no nutrients. So it's in crisis. What if we could induce in such an organ NDRG1 and enable this organ to have reduced cell damage because it is in this state of stasis? So that's where we hope to go in the future, and thank you very much for your attention.